Yes. আজকে জিজ্ঞেস ব্যাস একদম স্যার স্যার একটু জিজ্ঞেস করতে ঠিক আছে ঠিক আছে ঠিক আছে তো এই তো কথা। এক মিনিট এই লাইটটাকে জ্বালানো আরে
Good morning. A very warm welcome to our 13th Foundation Day celebration. We are extremely glad that today with us we have Professor G. Taru Sharma, the Director of National Institute of Animal Biotechnology, Hyderabad. And yes, we are very glad to have you here. So we will start our program with an invocation performed by our students, Oborna Sinha, Piali Mondol, Modhurima Chatterjee, and Antara Pal.
सर्वेषां स्वस्तिर्भवतु सर्वेषां स्वस्तिर्भवतु सर्वेषां शान्तिर्भवतु सर्वेषां शान्तिर्भवतु सर्वेषां पूर्णं भवतु सर्वेषां पूर्णं भवतु सर्वेषां मङ्गलं भवतु सर्वेषां मङ्गलं भवतु सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामयाः सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत् सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामयाः सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत् शान्ति शान्ति शान्तिः
I now request our Director in Charge, Professor Orinder Moitro, to deliver the welcome address.
Thank you, Anushwa. It gives me immense pleasure on this auspicious occasion of our 13th Foundation Day to welcome all of you to this event. In particular, for people who do not know, and the newcomers in the institute, as well as those who are in the audience and other places, we have 2 days that we celebrate with pride. One is our Foundation Day, that is today, which is 6th August. This is the day when the Biomedical Genomics Society was formed, the society that so far as the institute is in the ages of that society. And this is different from the Institute Day, which we celebrate on 23rd February every year. That is the day when the Union Cabinet approved of the formation of National Institute of Biomedical Genomics. So these are the story behind these 2 days that we celebrate with pride. And today we are really fortunate to have with us esteemed guest, Professor Sanjeev Jain from NIMHANS. I've been fortunate to know him for a long time, even from my days in industry, and we always look up to him for the kind of work he does in genetics of psychiatric disorders. Today we will have the opportunity to hear him on this Foundation Day lecture. I also welcome Professor Taru Sharma, the director of NIAB. She could manage to come in this short span of time, and welcome to the institute and to the Foundation Day. I also convey my greetings to our founder director, Professor Partha Majumdar, who is here with us, and my preceding directors, Professor Shanmula Sengupta, who is also in this room with us, and Professor Soumitra Das, who I see has joined online, is on the other side of the screen. I convey my greetings to the entire faculty, staff, students, and postdoctoral community of NIBMG and all the staff. This is an occasion for us to be proud of. This is the 13th Foundation Day that we are celebrating. We started our journey from the disused floors of the TB hospital nearby. And from there, we have traveled this much, covered many. In this short span of time, limited span of time, we have participated in many important programs, large events, and which are like notably the oral cancer and in SARS-CoV-2 genomics and many other areas of biology and human diseases. So without further ado, I would now hand back to Anusua for the next part of the event. Welcome everybody to this program. We hope the program will be a grand success, and we look forward to the next program which NIBMG will celebrate. So this is part of coming out of the pandemic, and in the process, we are very happy that we could actually gather today in person after a long period of time. Thank you.
Kindly request our founding director, distinguished professor and National Science Chair, Professor Partha Majumdar, to kindly introduce our speaker.
No. Oh, okay.
Good morning, everyone. It's really a pleasure to assemble here after a few years when we celebrated our Foundation Day online. And it's really very, very heartening for us to be able to see each other's faces, to be able to see the person who will deliver the Foundation Day address, in person. So I welcome all of you, and it's really a proud moment for me to introduce my friend and colleague for a long time, for many decades, Professor Sanjeev Jain, who is a distinguished professor and a senior professor and the Dean of Behavioral Sciences in National Institute of NIMHAN, National Institute of Mental Health and the Neurosciences. Professor Jain has been one of those very rare persons in India who is an MD in psychiatry, but has been completely devoted to understanding genetics of mental illnesses. I, for whatever reasons, have interacted with many psychiatrists in India in various institutions, various cities in India, but Dr. Jain has actually been very, very focused for many decades trying to understand genomic underpinnings of mental health illnesses. In particular, he's been studying schizophrenia and bipolar disorders, and as many of us know, that for these disorders, the spectrum of various kinds of the spectrum of the phenotypes are quite overlapping. So it becomes a challenge to first of all distinguish, discriminate between the phenotypes, and then to study the genomics of these different phenotypes. There is, of course, a lot of environmental impact on these phenotypes as well. So Sanjeev has been studying young onset, adult onset, and so on and so forth, those kinds of things to understand the impact of genes and also how it relates and interacts with the environment. Sanjeev Jain has also been very interested in the history of mental illnesses. So he's written quite prolifically in various journals and magazines on history of mental illnesses. He's also, one of the major impacts on mental well-being of citizens of India and Pakistan was the partition of India. And Sanjeev has actually edited a book, invited a large number of his friends to write, compose, edit a complete book on the impact of partition on the mental health of citizens, not just in India, but India, Pakistan, etc. So he's a very, very organized person, especially when it comes to documentation, documentation of history, documentation of phenotypes, and very careful documentation of family history, which is what allowed him or enabled him to understand genomics of various kinds of mental illnesses. So it's really a pleasure for us to have Sanjeev give us an idea about the challenges and pitfalls of his lifetime's work on mental illnesses. And we look forward to his lecture, his Foundation Day lecture, the 13th Foundation Day lecture of the Institute. I'm also very glad to have, in our midst, Professor Taru Sharma, who is the director of the National Institute of Animal Biotechnology in Hyderabad. So it's really very kind of her to accept our invitation and come and celebrate our Foundation Day jointly with us. Thank you very much, Taru. I would request everyone to put your cell phones on silent mode if you have not already done so. And I invite Professor Sanjeev Jain to deliver the 13th Foundation Day lecture of the National Institute of Biomedical Genomics. Sanjeev.
দেখিয়েছিলাম তাকে সোজা করে দিই। Sorry about that.
So, good morning, everyone. Thank you, Arindam, for inviting me, and thank you, Partha, for the over-generous introduction. But I'll try to explain in what the excitement of psychiatric genetics has been. It's obviously a challenge, held out lots of promises, and over the last few decades, all we seem to have are pitfalls. So it's really been a Am I audible? Yeah. So this is what we'll be talking about. Now, the urge to understand mental illness goes back forever. And so we have two ways of understanding mental illness. One is it a morbid phenomenon, which is a natural variation in all of us? Like some of us are tall, short, some of us more mad than others. So is it a normal quantitative variation of a normal trait that all of us have, like height? So it's a quantitative variation of normal psychology, or is it something completely abnormal that happens, like when people start having delusions or hearing voices, which is a very different thing. So that is a morbid phenomenon which have no analogs in a normal healthy state. As you can see on the left, these ideas of how to correlate brain anatomy, brain surface with various kinds of character is something that came up many in 1800s. But at this point in time, what we would like to see is how a particular chromosome deletion causes changes in cellular biology, into anatomy, and brain cognition, and finally in a psychiatric diagnosis. So what are the steps that transition from a basic genetic defect all the way to a phenotype of schizophrenia? So this is the kinds of issues that govern us, that drive us, because psychiatric disease remain, as we were talking earlier in the day, an island of medical ignorance. Now, mental illness is obviously very common, very prominent, and you can see a wide list of people from all spectrums of society, from Nobel Prize winners, James Joyce's 100th the publishing of Ulysses is being celebrated all over the world. And James Joyce's daughter had very severe schizophrenia. And he almost contemplated getting her leucotomized because leucotomy was a new treatment at that point of time. And of course, everybody in Calcutta knows Clive of Lassy, who basically ended up ruling India, winning the British Empire, but he had severe mood disorder and committed suicide. So he had a very severe recurrent manic depressive psychosis, and it's also debated whether Indian Empire was rooted in a manic episode, which allowed him to take on an army of 100,000 people with 10,000 people, grandiosity. And of course, he was acting on behalf of King George, who himself had bipolar disorder. So it's all kinds of, so mental illness has a way of intruding into our day-to-day life, into our day-to-day experience. And you can see all these actors from Robin Williams to Rajput, to Praveen Babi, to Virginia Woolf, all of whom have had mental illness. And of course, Shakespeare pointed out, great wits, that the partition between sanity and insanity is very thin. Over the last few years, we've also understood that given even these differences between neurology and psychiatry that have become popular over the last half a century are actually quite artifactual. If you look at it in a purely objective way, patients who have spinocerebellar disease, psychosis, frontotemporal dementia, Parkinson's disease, Alzheimer's disease, Huntington disease are completely normal till they are 20, 25 years of age. Absolutely, completely normal. And then for some reason, something starts changing and the illness has an inexorable deteriorating course. In some cases, it's a monogenic disorder. In some cases, and all the others, these are multifactorial polygenic disorders. But the ultimate clinical semiology is somewhat similar. And of course, they are located in different parts of the brain. But it's whether they are separate diseases or a continuum, because symptoms of one can occur in the other. Symptoms of psychosis are quite common in Parkinson's disease. Depression is very common in Parkinson's disease. Alzheimer's disease patients can have delusions and hallucinations. So there's no specificity to the whole process. All we know is that very slowly, gradually, the brain and the mind deteriorates. So this has become medically quite obvious over the recent few years, and this drives some of our work, as I'll show in a little while. So what are these disorders that we talk about? These are the mental disorders, that the ones with the star are the ones we are particularly interested in, which is OCD, psychotic disorders, conduct disorders, alcohol dependence, ADHD, bipolar disorder. These are fairly common all over the world. They are very, very expensive to treat, as the green bracket shows. Between addiction, anxiety disorders, psychotic disorders, dementia, these are the most expensive public health disorders to treat anywhere in the world. So they are common because they affect almost 5% of the population. Their symptoms, treatments, and outcomes are the same as anywhere in the world. These particular cost and public health burden are from Europe. But the small graph on the right shows the prevalence of these as per the National Mental Health Survey of India, and the numbers are exactly the same. So these cost in real terms, in terms of lives affected, the same as in India. And 1/3 of all treatment costs, of all healthcare costs, go to treating psychiatric illness in all its manifestations. Insights into the mechanism are very few, and there are very advances in the basic understanding are very, very paltry. Why are we specifically interested in this? The syndrome of schizophrenia is defined by periods of delusions, hallucinations, disorganized speech, negative symptoms, which are known as positive symptoms, followed by negative symptoms of amotivation, apathy, which can go on for months, years, or even a lifetime. But there are fundamental disturbances in cognitive function and the way abstraction occurs and the way concentration occurs. But at the bottom is the classic graph, which shows that monozygotic twins have a concordance rate of around 50%. First-degree relatives have much higher concordance rates for the same illness, and it falls sharply by the degree of relatedness. So while the general population has a rate of around 1%. So everybody has a risk for having mental illness, but the risk rises sharply the closer you have a relative. This is the same for bipolar disease. For a person with no mood disorder, related to nobody with a mood disorder, rates are around this. With one person with either a parent or a sibling with bipolar disorder, it jumps almost 2.5 times. And with unipolar depression, it jumps almost 15%. Another little bit is added. But for bipolar disorder, it comes down a little bit. So depression is defined as a 2-week period of change in usual functioning, of depressed mood, loss of interest, etc., and mania via the opposite. So these two disorders basically affect every aspect of a person's capacity to think, feel, or behave. So they change the entire personality for a period when the person is suffering from the illness. Relative risk for siblings, the simplest possible genetic model shows that the heritability in siblings is fairly high, 10 times higher than others. And this is much higher than that for asthma and hypertension or diabetes. And it's somewhere near this. Obviously, it's much lower than cystic fibrosis and Huntington disease. So the genetic roots of this, therefore, should be amenable as per the sheer calculations go. So what are we trying to discover? These are the basic rules of medical genetics. Is there an inherited component? If so, to what extent? What are important for certain kinds of disorder? Can specific gene variants be involved? What are the environmental influences that play on this? And how do they interact with each other? So a lot of our work has been driven by these 4 kinds of studies. And we'll go over some of these from our experience and from the others also. What is it that we need to understand? We know that it runs in families. We know that patients who develop schizophrenia may have subtle developmental anomalies in their brain. We know that a large amount of gene expression occurs prenatally, so that certain changes in the way the brains are being organized can actually be hardwired into in utero life. We also know that over lifetime, gene expression does vary. So brains, for example, dopamine receptors get expressed only after birth, and they mature in a particular pattern and they decay in a particular pattern. So there's a whole timeline of how gene expression changes in the adult human brain. Could these be multi-organ involvements? So patients with dementia ultimately die of cachexia because all the cells of the body start dying and they develop severe weight loss. And obviously there are growth deviations. There are many other issues of seasonality, age and effect, of neurodegeneration, how the brain, how the stem cell biology in the brain is maintained in terms of regeneration, neuronal modeling, and memory disturbance. And of course, the fundamental question which many students asked, are human beings the only species that have schizophrenia? So in which case it is a peculiar problem in biology that for all of the diseases you have animal models, but for psychiatric disease alone, you do not have an adequate medical model that limits the research to a considerable extent. No, I don't know if it's readable at all behind, but obviously any disease can be understood either in terms of the basic biology or its running or its occurrence in a family. So if it's a biology, it's very simple. You have Mycobacterium tuberculosis, you have a bacterial infection, you can understand the biology of the impact of that bacteria on the person. Also, you can track the family studies, or you can do more recently in terms of genetics an theoretical approach based on gene distribution invariants. So both these techniques are now increasingly powerful. So linkage studies had a very early start. In the beginning of the 1980s, the moment linkage methods became available, psychiatric disease is one of the first to be analyzed. From Icelandic pedigrees, North Scotland. So very early on, I think in 1997, Egeland and Gerling published a paper on finding a gene for bipolar disease in a 10-member family. And within 2 or 3 years, it had to be retracted because few more people fell ill in the family and the linkage scores dropped. So over time, it also showed you this whole problem that your diagnosis of an individual is not constant. Somebody who's normal today may not be normal after 5 years. And your linkage scores will need to incorporate that kind of dynamic diagnosis. Not everybody will remain well. So those linkage scores dropped sharply. So there was very little concordance between all the early linkage studies, and one could not find reliable linkage signals from sets of families anywhere in the world. So that created a problem. And over time, the amount of studies published on linkage findings in schizophrenia and bipolar disorder started showing the usual, normal distribution, a sudden enthusiasm and equal waning of interest. But that doesn't take away from the fact that very carefully a certain large families can actually be quite useful, because these need not look at population level information of minor alleles or allele structure and have the potential to discover certain alleles. So right now they're tending to come back in fashion. And now, for example, this particular graph is from a 100 plus member single family with psychiatric diagnosis, from which they estimate they can detect 105 copies of a rare variant of a private mutation if it does occur. So there's a worldwide effort to gather very, very large families. And this is one of the families with schizophrenia, bipolar disorder that they have identified. Now, we've been doing this for some years, and we've got almost 800 families now. But rates these are the basic clinical premises. For any psychiatric diagnosis, the rates of mental illness in these families were 2 to 10 times higher than the general population. So we do detect evidence of certain kind of accumulation of genetic risk. What we also observe, in most families, it's not the same syndrome which occurs. As Partha mentioned, sometimes index patient has schizophrenia, but the father has had a bipolar illness or the sister has some other kind of illness. So these diseases are not specific in the same way as classical medical genetics likes to look at a disease definition. There is a spectrum of symptoms and which vary over time. So in the families with bipolar disorder, only 37 families had the same diagnosis in all the family members, whereas 114 families had other diagnoses. So for all of them, you see that the blue bar is much higher than that of the others. However, in some families, especially with addiction, ill members all shared the same phenotype. So there is a core of so-called pure disease syndromes, but there's a big spectrum where the general risk for various kinds of psychiatric illness is inherited. Now, with all this, we now know with genetics that there are certain rare variants of high penetrance which could explain Mendelian patterns of inheritance. So single families with multiple ill relatives, and there'll probably be large number of variants of relatively small size for which you'll have to do something like a GWAS. So, as Partha mentioned, we've been working together for many years, and this was our very first attempt at linkage analysis. We collected around 30 families, and we did a whole genome scan using linkage markers. That's how I met Arindam. And with those big glass plates, and we started the very first linkage analysis. And so these were a small group of families, around 20 or 30 families. But if we confine our phenotype definition as only schizophrenia or as only bipolar disorder, the Lord score did not reach even modest levels. But the moment we combined all psychosis, we got a fairly robust Lord score of almost 3, which was then considered fairly good. It was a large region on chromosome 18p, which was fairly large, which housed these genes, GNAL, CIDEA, and PTP, and inositol monophosphate. And these regions have been implicated by other studies in the risk of bipolar disease. From that point of time, we did not have the capacity to drill down further into the same region to look at whether variation in these genes were important. But there were a few follow-up studies in the few years following publication of this paper, which basically confirmed these findings, or around the same time. We continue to collect families, and this is a particularly large family that we identified with more than 100 members, of which we had information on almost 71 subjects, detailed information, clinical information, of which 19 had bipolar disorder, 1 had a major depression disorder, 1 recurrent depressive, and 50 apparently unaffected individuals. And this is a small fishing community in Tamil Nadu who were one of them came to us for help, then he kept on bringing relatives. And because they were very interested in this, one of them was a psychiatrist. So he said that, let me get the whole family involved and let's see how much we can help you guys get this. So he got the whole family involved over time. And so we did a 10 centimorgan panel, and then we whole exome sequenced 11 of them. And we were able to find two single nucleotide variants within these 2 regions on 1q and 6p. Which were basically a variation in the CUNC4 protein and the cyclase-associated actin cytoskeletal regulatory protein, both of which are involved basically in vesicular retrafficking and sodium channel and in maintaining sodium balance inside vesicles. And the other one is a cytoskeletal protein. Now, this has been a prominent feature in neurobiology research, that the genetics comes up with biology that you had not previously imagined. We use drugs to treat psychiatric disorders which act on neurotransmitter receptors. For epilepsy, we use drugs that stabilize membranes. But many of the genes that are being found for either epilepsy or mental illness are structural proteins. So we really need to understand how actin cytoskeleton manipulates or regulates the behavior of membrane-embedded receptors and membrane receptor biology, which will require a very different kind of understanding. So the power of genetics to give you novel insights, which then have to be acted upon, is actually unique. Because coming from the clinic side, we would never have imagined that a cytoskeletal protein is implicated in the risk of bipolar disorder. So it's very, very important to do this from the ground up. Of course, the gene itself is completely conserved in all species. And this particular variant is shared by 7 out of 11 and 10 out of 11 affected members. So it's a fairly robust kind of a rare variant analysis. This is something we had observed many years ago when we were still doing gene-wise analysis. So this is work done in collaboration with Professor Brahmachari's lab, where we found lots of individual patients and families who had rare variants, either in the synaptogyrine gene or in the MLC gene, that's a leukoencephalopathy gene. A rare variant had been discovered in Germany, and then when we sequenced the gene, we found a couple of other rare variants. Then we looked at the literature, there are a number of rare variants correlating with psychiatric disorders. But these could not be followed up, and also because soon after this, when we did the IGV data, we found that some of these rare variants were actually common in other populations. So the whole premise of this got slightly muddied. So and at that time, since we were doing only gene-wise analysis, we could not go very further. For the last few years, thanks to the DBT, we've been engaged in slightly more advanced studies where we collect these large families and do a whole exome analysis. So this is just 2 of the families. There are about, this particular paper describes 40 and the next one describes 80. So we have a large number of families with at least 3 or more people affected with a severe psychiatric illness. So not just 1 or 2, we had to have at least 3 or more people affected. The 3 diagnoses can be schizophrenia, bipolar disorder, OCD, dementia, whatever, because we assume that there is some kind of a shared heritability between these disorders. And all of them have been subjected to exome sequencing, and marked on this map are the various genes that we find. And I'll be describing that in a bit more. So a large number of the genes that we detect are genes which are actually implicated in syndromes of neurodevelopment, which have already an OMIM classifier attached. So as you can see, spinocerebellar ataxia, myoclonic epilepsy, Joubert syndrome, Cornelia de Lange syndrome, number of them in Joubert and Meckel syndrome. The mutations that we detect are obviously run through the whole pipeline of being deleterious by at least 3 or 4 predictive mechanisms. They should not be conserved. They have to be rare variants present at a particular frequency, below a particular frequency. These are all rare variants, deleterious, shared between members of the family. So they seem to be having some signal, but they don't overlap exactly with the known mutations in the gene which cause these full syndromes. So our assumption or a hypothesis is that subtle variation in genes which otherwise cause a full syndrome, call these attenuated syndromes, which increase the risk for psychiatric illness because of the way the brain is being developed or being organized, but they are not confirmed, they are not fully penetrant in that sense. So pleiotropic kind of effect is likely. So these are the various processes, and as far as processes go, they go back into processes that we understand about psychiatry. G protein receptors, cortex cell migration, calcium channel signaling, and postsynaptic membrane potential. These are processes that we know from other biological research, are processes which are definitely altered or dysfunctional in psychiatric disorders. So we think overall, they tend to point towards a convergence of biology and organ involvement. So this is a further paper. This is done with the help of Dr. Samsiddhi here. This is still under review. So now we have 80 families in which we find 67 single nucleotide variants in unique genes. About a third of them are in, like the earlier paper, in OMIM-related genes. And as you can see, these are segregated with SMI. The variants are not pathogenic in the primary OMIM syndrome. Most are private to the pedigree. Now, this confuses us a lot because this becomes like clan genomics. So if a particular family, at some point a single nucleotide variation has occurred, then, and if there hasn't been much admixture, that pedigree or in that particular clan, that particular variation, like this particular syndrome, as you can see, schizophrenia, bipolar disorder, substance use, OCD, all of them have clustered in the same family. relate to the occurrence of a Gemin5 mutation, which is otherwise linked to cerebral atrophy and motor dysfunction, NETCAM gene. But there are so many people affected here who carry the same variant. A number of variations were, and this is a routine QC of that, what are the total number of variants we found, and more or less they are, I mean, our experiment was in keeping with whatever trends there are. We also looked at it in terms of syndromes, and as you can see, many of them are involved in both neurodevelopmental and neurodegenerative processes. So spongy degeneration of the CNS, developmental disorder, developmental disorder, hereditary spastic paraparesis, epilepsy, growth retardation, hypoplasia, autism, etc. So there seems to be some kind of convergence that this kind of mental illness may actually be subtle changes in neurodevelopment, which are then priming the CNS. We also looked at how, if we are detecting so many of them OMM syndromes, if we look at other organ systems, do we still get a signal? And we found that we only get an OMM signal for the head and neck and brain development, and not for or other rest of the body. So that we also show that the signals that we are getting are specific to the development and functioning of the brain and associated parts of the body. By this time, the Gulsner et al. Had looked at a series of single nucleotide variants in a defined set of genes, which they call broadly synapse-related genes, in the Xhosa community in South Africa, which is a native South African population. And they had reported that in schizophrenia in general, impairments or changes in the broad group of proteins that are linked to synaptic biology, 1,200 of them, occur more frequently than by chance. And we wanted to see if in another non-African population would we detect the same thing. So we ran the same list of genes using the same kind of analysis, and synaptic genes come up as being significantly more likely to carry rare variants in the synaptic function genes. These did not overlap with the neurodevelopmental genes. So now we have two kinds of scenarios going on. There is some kind of rare variant which is being inherited in combination with certain things that disrupt synaptic signaling. Probably it's a combination of the two that ultimately results in a psychosis, but which is the chicken, which is the egg, which is primary, which is secondary, we have no way of knowing. But these kinds of processes that we are talking about, and the processes involved are formation of presynaptic membrane, postsynaptic membrane, all these things have high rates of single nucleotide variants. So ultimately, it boils down to the fact that, as we keep discussing all the time, if you look at the epidemiology of rare variants, the rare variants which are medically relevant often tend to have a disproportionate representation in diseases of the brain, of the CNS. So that is an empirical finding from genetic studies all over the world. And so rare variants, the symptoms and syndromes often overlap. And if you look at the expression patterns of all the rare variants that are detected in brain disease, then you find that the symptoms of brain disease also tend to overlap. So from pyramidal signs to dystonias to behavior problems to motor function. So symptoms also tend to overlap. There's no single module here which will overlap directly onto a gene. So patients can have combinations of these symptoms over the lifespan, and therefore the genetic basis may also tend to be slightly more confusing. In general, if you look at all protein coding genes, and you look at the genes expressed in the brain, which are known to be linked to autism or infectious or developmental disorder or epilepsy, and therefore by extension to most other neuropsychiatric disorders, these genes tend to be larger. And since we know, and they have richer GC content, and since we know that a number of so-called mutations or variants are a function of gene size, it's quite likely that genes of the brain therefore tend to accumulate more number of variations or more common number of genetic variations. So we would really have to look at this in much more detail over time, because this difference is quite marked. All protein coding genes and brain disease linked genes. There's a distinct shift. And so it may really be a function of way evolutionary trends have shifted certain brain expressed genes to be larger, more complex, and more GC rich, and thereby inherently have more chances of mutation. So this particular family exemplifies that. So this is one particular family in which all children are affected. All 6 of them have an illness arising from one father. The father was ill and all the 6 children are ill. But between them, there is not one single rare variant except the first one, which is shared by all of them. So even if you did some kind of biology, leave one out kind of analysis, how would you actually go about it? So in many of them, now we have iPSC lines, we have neural stem cell lines, we have the MRIs, and we have the whole exome data. So now you can imagine building up experiments at different kinds of biology to tease out each of these. So we know the pathways involved, Notch pathway, MAPK pathway, PIP3 pathway, but many of them are involved in neural migration, cell migration, cell migration, cell migration. So now you're getting convergence to processes that underlie the basic biology of the brain. And that is what we can investigate. Now, of course, over the last, around the time that we started working together, we didn't have the kind of wherewithal like all the fancy toys we have now, or at least you all have now. But so we were doing basically candidate gene studies, looking at RFLP at a time. So we did all kinds. We could guess candidate genes, would still require, if you did it properly, would require millions of SNPs to be tested. We can guess possible gene from what we know about mental illness, and we spent a lot of time doing that. The drugs used in psychiatry or Parkinson's act through dopamine pathways. So let's look at everything in the dopamine pathways. We know serotonin controls mood, so let's look at everything in serotonin. We know that postpartum depression occurs. Let's look at everything in oxytocin-related pathways. We didn't find anything reliable, because what we thought we knew, we don't actually know. So all this became the graveyard of molecular genetic biologists, that whatever theories you had about molecular biology became completely discarded. So now we start going to indirect GWASs, which basically ran on linkage disequilibrium maps. This is a portrayal of all the graveyard studies that we did. We must have published around 20 papers, small sample sets of 300 to 1500, other than ApoE4, which consistently stood the test of time. Nothing else really stood the test of time. So, but that's not really true, because even for DRD2 receptor, we find certain pharmacogenomic markers. For COMT, we do find aspects of biology, and serotonin transporter, we do find certain aspects of biology, but by no stretch of imagination could the link with disease itself be confirmed. Even for DISC1. DISC1 is the classical disrupted in schizophrenia 1 pedigree, which was reported way back in 1960s, and then worked up, gene identified sometime in the late 70s or mid 80s, and has been worked on endlessly since then, because 14 people in the same family have carrier DISC variant. But we could find some partial overlap between the SNPs of DISC1 variant, which were overlapping the Scottish samples, but none of these were otherwise significant. So we can now start doing genome-wide studies, and we can try to build up large samples. But the problem in India, of course, is that sample sizes are difficult to actually get. This rely on the basic principle that the risk allele arose sometime in the past, and by patterns of just vertical transmission, it is widespread in the population. You get incomplete penetrance, you get disease phenotypes sometimes, you get non-genetic factors or interacting loci at various genes, and you get minor contributing alleles. So you need to understand all these scenarios when you collect a large sample of common disease. Like I said, these diseases occur in 5% of the population. So these are common disorders. But at the ground level, when you collect data, you can have all these scenarios occurring simultaneously, which makes genetic analysis slightly more complex. So over time, what we've discovered is that there are a large number of rare variants which run in families, like the 22q deletion syndrome, various kinds of rare variants which are being identified, including some of ours. But you also get a large number of small variants of small effect sizes. And to detect these has taken a huge impact, has a huge effort. Number of cases as they have climbed, and I don't think the slide is visible at the back at all. So from the first association study way back in the early 2000s of the signals, as the sample size improved, you started detecting more and more signals. And over time now you have more than, by the time you have the second psychiatric genomics consortium, you found 128 common variants with predominant effects on excited neurotransmission, that's glutamate receptor, and 22 common variants which have cumulatively a very small effect earlier. At the same time, when you look at whole genome sequencing, the whole exome studies, which less than 100 trios, over time, by the end of the second decade, you can find that there are a number of genes which are being implicated purely on population kind of studies, like say D1A. And the polygenic burden of these genes is again implicating the same kind of biology that we discovered, that we were talking about earlier, voltage gated calcium channels, etc., etc. So ultimately, whether it is GWAS or the rare variant studies, they are converging on particular processes in the brain, relying on the control and regulation of neurotransmission and neuroplasticity that are being implicated in the psychiatric disorders. Now, what is worrying now? Now, obviously, by now people have realized that these kinds of diagnostic splitting apart may have its utility in the clinic, but genetically speaking or biologically speaking, you may need to merge data sets because there may be a general liability for disease. It's like the issue between diabetes and hypertension, which is a metabolic syndrome. Do you take very precise phenotypes or do you take a broader phenotype? So all of these syndromes listed, which are relatively common syndrome like schizophrenia, ADHD, OCD, substance use, they have really, so the size of the signal has increased dramatically as the number of cases have increased. Bipolar disorder is still lagging behind, but schizophrenia has really given rise to all kinds of information as inflammatory bowel disease. But what worries me at the end of it is that the signal for height is much the same for schizophrenia. The genetic basis of height is the same as schizophrenia. Now, that is very confusing. I can make out a tall person very easily, but I can't make out a mad person so easily. So what is the difference between the trait of height and the trait of mental health, which makes, as far as the genetics are concerned, they both have equal heritability. But in the phenotypes, the concept of variation in height is easy to understand, but a variation in the notion of sanity is very difficult to understand. So what is it that we are talking about here? So what we get over time is that we get a number of loci for schizophrenia, we get a number of loci for bipolar disease. The sample size go up into thousands and thousands. And as you can see, schizophrenia overlaps strongly with bipolar disease. Bipolar disorder corresponds largely with schizophrenia. And Tourette syndrome and obsessive compulsive disorder go along with each other. Tourette syndrome is a complex basal ganglia disorder in which people have tics. And during the tics, they also have mental tics. So they are forced to repeat the same thing over and over again. So theoretically, you can easily club them up into shared biology of the basal ganglia frontal brain circuits. For some reason, this is occurring together. So it's all very nice. What is troublesome is post-traumatic stress disorder with schizophrenia. So it tells you that social factors or social stress is also playing a role in the manifestation of schizophrenia, even in terms of genetic risk for experiencing post-traumatic disorder, which is otherwise a purely environmental disorder. I mean, only if you experience trauma do you have a post-traumatic stress disorder. But to develop a post-traumatic stress disorder syndrome, if you do a GWAS, the findings overlap with that of schizophrenia. So in addition to the so-called non-trauma induced schizophrenia, now there's an overlap. How stress, the experience of stress or the way the brain or mind responds to stress may be a pathway which is also linking back to schizophrenia. So the gene environment issue therefore comes alive all over again. It's not merely neurodevelopment or neurodegeneration. It is also how the brain is handling stress that is implicated in the risk of schizophrenia. Now, this is not true just of bipolar disease. And sorry, I should have mentioned one thing earlier. What is also interesting is completely counterintuitive: attention deficit hyperactive disorder and educational attainment. Now, this is all UK Biobank study, and so I don't know if the British school system is very different from all of us, but for some reason they find educational attainment as being a measurable genetic signal. And some of these overlap not just with ADHD, but also with autism. So educational attainment or the capacity of the brain to handle useless information, which is what education basically is also linked to certain kinds of risk. So we can test overlaps between all these syndromes, between ASD, ADHD, bipolar disorder, and you find that distinct overlaps between ASD, bipolar disorder, and schizophrenia. And not only that, when you look at gene expression patterns in different parts of the brain, in prefrontal cortex, and the skin and the liver, the expression patterns in the cortex correlate heavily with the genes at risk, whereas the rest of the body doesn't. So we know now with all these genetics that these genes may be implicated. They do affect the biology of the brain. They do affect biology of the brain in all these syndromes in a congruent manner. And therefore, there is likelihood that these disorders are actually joined at the hip. And in particular combinations and in response to a gene environment, particular groups of symptoms develop. But as syndromes by themselves, they probably do not have any heuristic value. So which is what we come to, that these disorders, that all the disorders that we talk about as the 5 major disorders, OCD, bipolar disorder, schizophrenia, are all linked to each other. So, and as you can see in the graph, schizophrenia, bipolar disorder are very strongly linked. At the other end, obviously, you find that autism and attention deficit disorder are inverse to each other. Children who are shy are not likely to be risk-taking, aggressive kind of children. So that's commonsensical. But for the large majority, there is a huge overlap between the signals of one syndrome. Now, obviously, they suggest that there's a kind of polygenic overlap, but the studies in the European populations this is a classic paper published around 10 years ago when the first Wellcome Consortium and the PGC program started, that the polygenic risk scores dropped sharply when they tried to include Hispanic Americans and African Americans. From p values of 10 raised to minus 22, it dropped down to 10 raised to minus 13. So a huge fall. Now, given that, people are very worried that would these findings be applicable to the universal population, or would they be population specific? So recently, the people have attempted the correlation between European and Asian population. By Asian, they mean Chinese. I think only about 3% of the PG Psychiatric Genomics Consortium has got non-European ethnicity, which is basically Chinese. No other country has been involved till now. So the allele frequency of these genetic variants between East Asian and European population, 70% of them, 70% of the allele frequency of the schizophrenia-associated genetic variants differed significantly. However, the mean allele frequencies did not differ between the populations, so you could still get roughly overlapping risk factors for schizophrenia, even between the Chinese and the European populations, which really gives you some idea that these diseases may share a fundamental backbone, which is similar, which need not be contaminated or confounded by the amount of variation we see within the human species. As an example, this is the highest, the potassium-gated voltage-gated modifier subfamily, which has the lowest value in all the GWAS, and the highest, which was the PCC enzyme, which controls cell division. And you can see this, the allele frequencies of this highest value was more or less constant across the world, whereas this one was also fairly constant. So you find that even at the extremes of these risk genes, their allele frequencies are not too far apart. Of course, many years ago, with the Indian Genome Variation Database, we had observed a similar thing with two completely different, this is not to comparison, but this is the COMT and the DRD2 alleles, which we showed that the differences between comparing the Indian population to European and Japanese and the Far Eastern showed clusters of Far Eastern prevalence here from the Han Chinese and from the European towards this side, which is obvious by the geography of India. So there was presence of this kind of variation, but the effects of the COMT and DRD2 we don't know at the moment. So at this point of time, we now realize that the relationship between the 8 psychiatric disorders, bipolar disorder, schizophrenia, major depression, autism spectrum, ADHD, Tourette syndrome, OCD, and anorexia nervosa, can be roughly classified into exactly what we were talking about in the beginning of psychiatry 100 years ago. There are mood and psychotic disorders which are qualitatively different. There are early development neurodevelopmental disorders which cause abnormalities, and these are disorders with compulsive behaviors which are adult onset. So ultimately, it still boils down into modules of how the brain-mind works. that there are things which regulate your thinking and your mood. They have a series of genetic variations. There are things which alter the way the brain develops, and there are disorders with impulsivity, with like food, like eating disorders and compulsive disorders. So many of them overlap with each other. And if you look at it in more detail, again, I'm sorry, this is not being projected at all. So there are a number of GWAS signals which differentiate between schizophrenia and bipolar disorder. There are certain things which are absolutely common between them. And when you differentiate these patients, you find that bipolar disorder who do not have delusions or hallucinations are somewhat different from the schizophrenia, and the increasing the amount of schizophrenia risk genes increase the risk of experiencing psychotic symptoms. So it's not the syndrome which is important, it's the symptoms. How likely are you to have delusions and hallucinations is being inherited rather than a syndromal diagnosis. So there's something fundamental to the biology of the propensity of the brain to develop odd beliefs or delusions, which is being inherited. So these kinds of data is also now becoming clear. So now we get, now if you combine this variant analysis like we have done, we also find that there's a marked increase in the occurrence of rare variants with the sequencing platform. So over the last 4 or 5 years, the number of rare variants with extremely rare variants being linked to schizophrenia has actually increased. Now, this looks very fine on a graph, but this is data from 25,000 or 30,000 whole genome sequenced individuals, and they find about 40 people who have this variants. So the cost and the effectiveness of doing this on a large scale, and given the discrepancies in rare allele call rates between populations, this really needs to be understood in slightly more complexity. So of all these, we find that all these genes, whether it's 3q29 deletion or SETD1A or GRIN2A or trio genes or the calcium channels, all of these start accumulating with the minor allele frequencies are in the range of 1 in 100,000 or more. So how will you actually plan this? And at the same time, if you look at the various kinds of variants that we get in the human genome, you find that protein truncating missense variants are occurring much more common in schizophrenia and bipolar disorder. Missense variants, and at this time, at synonymous variants is absolutely the same as baseline as the rest of the population. So there is an accumulation of protein truncating missense variants in schizophrenia. At the same time, there is also a problem that they also have high rates of de novo mutations, in which case all the segregation and familial patterns become useless. So trio studies have now shown, again going into hundreds of thousands, that there's a high occurrence of protein truncating de novo variants, which are occurring again with schizophrenia, which are very high. In 52 genes, you get a slightly higher rate, significantly higher rate of protein truncating variations or missense variants, and synonymous variants are occurring at exactly the same rate. Now, what is driving these high rates of de novo mutations is something that we don't understand. It's now estimated about 3 to 5% of children with autism and early onset psychosis actually have de novo variants. And whether that is an inherent part of new variants being generated in the human species, we have no idea. But there seems to be an increased risk of this as a problem in our sample sets. Now, that's as far as commonalities are concerned at the genomic level. At the clinical level, we've also observed that there are certain differences. For example, in India, in bipolar disorder, people have more manic episodes, which we've published in 2 papers. Schizophrenia generally tends to be somewhat milder in non-Western countries. And mortality rates are also slightly different, as has been observed at NIMADS. On the other hand, we do find that there are certain subtle genetic differences, not neurogenetic, but genetic differences. So we know that haplogroups for Huntington disease in India broadly overlap with the European sample sets. We can look at north and south ancestral origin of Friedreich's ataxia, completely the independent mutation events, and for lots of variations, for lots of genes, we get a lot of variation within the human species. So we've been trying to look at data of looking at GWAS kind of data. So this is work that we have just begun. So this is the data on the first 1,000 subjects, 1,000 or so subjects, South Asian subjects, as red triangles here. and the European data set is here. So overall, our data sets seem to confirm that for the vast majority, our sample sets are not very different in terms of the PCA plots between those of European ancestry as far as medically relevant GWAS things are concerned. Of course, you do get a lot of variation within India, between north and south of India, but the variation between north and south accounts for about 3% difference in allele frequencies, whereas in the whole of Europe, it accounts for between 1 and 2%. So it's slightly larger than Europe, but it's not astronomically larger than that genetic variation between, let's say, between Scandinavia and Spain. Is the difference between Punjab and Telangana is perhaps half a magnitude higher or something. So we think that we should be in a reasonable space to conduct equivalent GWAS studies without worrying too much about population substructures at this point in time. Why do we believe this? Because we map the minor allele differences between European and South Asian populations. My colleague, Dr. Hola, found this interesting way. We looked at the European minus South Asian frequency at a sample divided by European plus SAS. So in the middle are those small line is the ones which are common, and these are the ones which are more in the South Asian, and these are the more in the European. And as you can see, the vast majority of alleles lie in this plus minus 0.5, so that you can use these fairly effectively for doing a GWAS, because they're not, they are obviously they'll be different, but as long as they're within that range, you should be able to do GWAS yourself. And the normal distribution of transdiscrete BMI by gender was just this much. So again, we could find that these were roughly similar. When we looked at the minor allele frequencies of the various traits that have been looked at for cholesterol, schizophrenia, OCD, neuroticism, impulsivity, height, etc., most of them are within this range, within the working range of plus minus 0.5, actually within 0.3, except for hair color, eye color, and alcohol dependence, which we do know varies a lot between Europe and India. So more or less, the traits that we are interested in are lying within this region, so that we should be able to look at how these traits are, how the polygenic scores in one ethnic group translate to the other and the other way around. Of course, we are also interested in how, and again, the question came up earlier in the morning with the students, that how much of it is evolutionarily derived. So there has been a lot of interest in how the archaic introgression of various components or copy number variants has changed the impact of certain behaviors or genes in the human. And we know that recently evolved genes have involved human-specific methylation regions, which are specifically enriched in the risk of schizophrenia. So as our society, over the recent, let's say, the current Homo sapien compared to Neanderthals, have certain positions where human-specific methylation regions seem to be more specific. And this we know from the genome variation, from genomation, from the 1000 Genome Project, that there is a large amount of the amount of Neanderthal genome in all the societies is slightly different. Coming to more proximate issues, we know that the outcome of mental illness is very different in different parts of the world. And in India, for example, the famines of the late 19th century resulted in a 50% reduction in those with mental illness. 50% of those who were ill died as between the census of 71, 81, and 91. So you know that famine and natural selection processes like infections and famines reduce the numbers of mentally ill. So can we detect those kinds of signals that we are talking about? So this is work done with Mayukh Mondal, who was introduced to us by Partho. So basically, mental illnesses have two basic issues. Evolutionary paradox that the rate of mental illness has remained constant throughout human history. There's never been an epidemic. There's no society who's free of mental illness. So it's a constant rate, and unlike all other illnesses, or like we say we are undergoing a diabetes epidemic or a COVID epidemic, this is a constant rate. So what is the reason for maintenance of that constancy? Why is there no selection pressure against it? And antagonistic pleiotropy, previous selection may bias towards certain alleles associated with late onset disease. Late onset disease, which occurs after reproductive whatever has been fulfilled, will theoretically have no selection pressure because the genes have already been passed on. So there's no specific selection pressure against late onset disease, and many psychiatric diseases are late onset diseases. So we took one member each from all these 80 families, from the 80 families that we have with multiple people who are ill. And we did a population branch statistic to investigate signals of selection. We found 74 genes under positive selection, and 20 of these have been previously implicated in genes linked to schizophrenia, Alzheimer's disease, and cognitive abilities in GWAS. So you then get a sense that brain-related genes have actually been selected for, but immune-related genes showed the greatest evidence of selection, interface with infectious disease and immune mechanisms, because we know that what we call immune processes like cytokines are actually neurodevelopmental modulators also. So astroglia are the immune crossover between brain development and brain maintenance is actually quite high. ApoE4 protects against viral hepatitis, against diarrhea in the childhood, but increases risk of dementia as you grow older. So there may be all kinds of evolutionary paradoxes that are there in late onset neurological disease, which will account for its constant prevalence in Homo sapiens, especially as the populations become older. There's no way that you can eliminate this, because for that you would have to eliminate almost half of our genome. So ultimately, what is it that we are talking about? We assume that there are genes at play here. The genes can interact with early life infection or trauma. We know that viral infection in pregnancy increases risk of schizophrenia. We know that infancy and pre-adolescent, there may be functional impairments. Children may be schizoid, may be kind of quiet, or may be impulsive. Then at-risk mental state develops, which is poor social integration. And within this, if there's things like cannabis use or extremely traumatic life events, you get a psychotic crisis, which occurs and relapses depending on booster hits. Now, this becomes a lifestyle perspective. The genes are there from here, but the way they'll express becomes a function. This becomes important also because in terms of biology, what you understand is the perinatal and prenatal development of the brain itself, which is being studied. We know that myelination, interneuron maturation occurs in spurts during early childhood, infancy, adolescence, and middle age. But all kinds of psychiatric disorders, the onset of schizophrenia typically occurs between 15 to 25. So you have this window when the brain is making new connections, pruning old connections, learning new things. At that phase, the brain is somehow more susceptible to develop severe mental illness. And once we change this, once it develops, what happens is to the mesocortical dopaminergic projections or the glutaminergic synapse density becomes altered lifelong. And how to reverse these with drug treatment or by social interventions or by rehabilitation becomes a series of interventions. To put it in another way, this is the path of the brain as it develops from childhood to old age. And as you can see, there are significant changes in brain structure over a lifetime. So we decided that because we can't compete with the GWAS, let's study our subjects in more detail. So what we find is that if you look at this data from 250 subjects. So across these diagnoses, there is significant atrophy of subcortical networks, irrespective of diagnosis. It is slightly specific for autism disorder in terms of anterior dorsomedial nucleus and the frontoparietal pathways. In schizophrenia, the posterior pathways and the caudate and the basal ganglia. And for bipolar disorder, again, subcortical and sensory motor and cerebellar systems. So you know that certain parts of the brain are getting more damaged when the illness progresses compared to normals. Now, in one particular family, for the big family I showed you with multiple people affected. Now, here you find that cognitive functions are more significant, are quite damaged in both the individuals. New learning ability was more impaired in the younger person, who was actually much, and there's significant widespread atrophy in this person, who was the older person, compared to normals. So therefore suggests that there is anomalous patterns of pruning which are happening. The brain is actually getting atrophied in terms of synaptic density much faster in people who have a mental illness. Now, we are still to get the genome data for this, and that is what we will be trying in the next few months to get done with the help of an IBMG. But what we have is a proxy marker. The greater the number of family relatives, first degree relatives, brother or a sibling with mental illness, the higher is the lower is the volume of the hippocampus, amygdala, and insula. So now you have a factor where, you remember we started with families who have at least 3 people affected, who therefore presumably have more greater genetic risk. Now we find that that risk of more family members correlates with smaller brain volumes of particular parts of the brain. Now, so our next effort will be to identify what are those genetic factors that actually correlate with this altered brain structure, especially of the subcortical and limbic system structures. Now, obviously, there are, like I said right in the beginning, there's gene environment issue. So when you look at these families, because these are families which are assessed in great detail, you find that 2/3 of the sample have experienced adverse events in childhood, either sex abuse, physical violence, basically sex abuse and physical violence. This seems to bring forward the time of onset of psychiatric disease. So even in the presence of genetic risk, if there is excessive occurrence of childhood trauma, the illness becomes earlier, starts earlier. This is specifically true of alcoholism and OCD, which is much more prominent. But for bipolar disorder, the risk that what brought forward time is not so much. So even those with low genetic were related to age at onset, and high genetic loading was anyway linked to early age of onset. So between the two, there is an interaction by which family environment affects this. Now this becomes a chicken and egg story. If you have a parent with mental illness or with alcoholism, the chances of the family experiencing stress is very high. There'll be violence, there'll be all kinds of unusual behavior and social stigma of various kinds. So all these factors can combine with each other. Now, when we look at the neuropsych traits, which is the usual things of processing speed, cognitive flexibility, working memory, new learning and recall, and social cognition, these are the patients, these are the first-degree relatives, and this is the normal population. You can find that first-degree relatives often lies somewhere between the two, so that there is a gradient, like a quantitative trait, which seems to correlate with the degree of risk. Now, so therefore, if you remember right in the beginning, I said, is it a normal trait or is it only a condition? So now we are finding that there's a trait kind of quantitative trait inheritance, which makes the relatives be halfway between normals and the actually ill patients, except for the domain of social cognition, where the relatives are almost as impaired as the patients themselves. So this also accounts for many other theories of mind and many other cognitive psychology theories, but that I won't have time to go through these days. So what are we talking about? We have deficits in cognitive flexibility, working memory, first-degree relatives also show a change, and the extent and range of trait dysfunction and structural and functional differences in the brain are what we'll be looking at now. Ultimately, we want to understand the aspects of our genome in a way that understands not only why we are ill, but also who are we, how we're related, and why differential responses to drugs. Because ultimately, we want to understand psychiatric disease both at the economic and environmental dynamics, but also at genomic substrate. So we have to, as a discipline, psychiatry needs to understand all this, because all you get to study is human action and behavior. And within this, how we can do this further. So the next few slides will show only that, and then I'll end in about 10 minutes. So this is looking at epigenetics. So we looked at children of alcoholic fathers who had high adverse childhood events, had high prenatal stress scores, had high externalizing symptom scores, high ADHD and conduct disorder. These children, when tested by giving a small reading task, and you measure salivary cortisol to see how the stress response occurs, and you find that children of alcoholic fathers had a slightly anomalous and a longer elevation in their cortisol levels. So there was the cortical response to stress was blunted, but different from that of controls. And this depended entirely on the amount of, now this correlated well to hypermethylation of the serotonin transporter gene, which we otherwise know modulates anxiety. So now you have a mechanism. Of course, this will need to be expanded in much more detail, but at least you are beginning to understand how early childhood stress alters methylation patterns, which in the presence of particular genetic polymorphisms can predispose to other kinds of depression, etc., etc. So now we can start connecting the dots much better than what we were able to earlier. Because you do know, if you remember that PTSD, etc., that altered stress responsibility may affect vulnerability to adult psychiatric disorders. So unraveling this kind of environmental stressful response is therefore very critical to our understanding. This is a completely different thing done with colleagues again in Calcutta. We've been looking at nutrition, pollution, and poverty and impact on brain function and of stunting. So all these effects are quite remarkable. And how this interact with genetics is again something that will come up when we get the data back from these people. So this is going back to the same slide of all these pathways. And I don't know if this will run. So you can, this is the left side is the control, and you can see the cells moving. And this is from the patients. The idea is that this gap should be covered up. So what we find is that the time taken to cover this gap between control lines and lines from this patient with bipolar disease is somewhat different, and the paths that the cells take are somewhat more chaotic. It's much easier to see here. So this is the this is data which we analyzed with the help of colleagues at the Indian Institute of Science. Those of you who have seen birds flying or fish swimming, they tend to swim in herds, and there's order to it. So it turns out that the cell migration from patients is inherently more disordered than that in the normal lines. Now, what is the generation of that order versus disorder is something that we don't understand. We know the minds are disordered. We didn't expect to find cell movement being disordered. So looking at the metaphors can be actually quite useful. That is, does disordered cell migration or cell biology actually underlie some of the migration defects, which we then found in the genes which are involved in cell migration, producing risk for psychiatric disease. So now we've been able to take this further in many ways. This goes on to much more fundamental levels also. For example, the cell viability of patient-derived cell lines in response to lithium. So we know that lithium stimulates cell division and is a cell protective agent, but we find that patients who did respond to lithium showed this response to lithium, whereas those who did not respond to lithium did not show the effect. So now you have a cell culture experiment to correlate with the clinical response to lithium, whether a patient responded to lithium or not. Now, what is the biology of this process? Why the mitochondrial potential was different in patients, and otherwise we don't know. So to close, mental illness can have various kinds of outcomes. Kurt Gödel, of whom all of you know, died of paranoid schizophrenia because he developed this delusion that if food was being poisoned. He was at Princeton Institute of Advanced Studies. He locked himself in a room. American laws prohibit you from forcibly feeding people, so he starved to death. This is the average. This is the outcome of a person in India kept tied to the bed because they can't afford treatment. So same illness. Kurt Gödel and this person have the same illness. Outcomes are very different. Kurt Gödel gets a Nobel Prize on the way. This man gets out of the bed only when he's brought to hospital. Otherwise, he's kept tied. Brain structures, brain activity, EEGs, everything we can study. Okay, and this is obviously something that all of you are familiar with. It's part of your exhibition, that nothing in biology makes sense except in the light of evolution. And since medicine is the applied biology, one has to apply those principles to medicine itself. It's not enough to think of medicine as some extra field. Medicine is nothing but biology applied on scale, on the human scale. Biology itself is much larger. So we have to understand human disease across all these things: patients, normals, humans, animals, living, etc., etc. And the commonalities is what we need to discover. So what is it that we have to introspect about? We have traveled far in these 20 years, but the problem is that in psychiatry you have this endless doubt about your own discipline. Am I really madder than my patients? It's a fundamental doubt. So it's a very fundamental doubt. So there may be no differences between patients because till now, for 200 years, we've not been able to discover a single biomarker. There's no test. You can't go to Lal Path Labs and say, do my test and tell me I have diabetes, I have this, I have that. Nothing like that works for psychiatry. So there's no marker. So perhaps it is just an inherent variation of species that we are talking about. Evolutionary terms, an error rate of 1%, you can live with. 99% of brains work perfectly well. Godel works perfectly usefully. It doesn't matter if at the end of his life, he had this problem. So the actual disability caused by these disorders may be adding to the variation of possibilities of brain emergent thoughts and ideas, or the urge for creativity, like that first slide. James Joyce, Albert Einstein's son who had schizophrenia, Heisenberg's children and grandchildren who have schizophrenia, and all that. The disease differences may be too subtle or specific. Like we were talking yesterday, the problem with brain disease is that everything happens inside the cell. If the cell membrane broke, like happens with cirrhosis or liver disease, you'll get enzyme leaking out, you'll get a biomarker. In these diseases, the cell membrane never breaks. There's no widespread cell death. It's a very gradual cell death, and our existing technologies can't image that. So we have no way of knowing how this process is occurring. There are population differences. So certain populations, like we see with Huntington disease, there are vast population differences, 10-fold or 20-fold difference. But for schizophrenia, it's roughly the same. So why this difference in brain disease? And evolutionary, of course, as I've been talking about. So there's some light at the end of the tunnel or the beginning of yet another tunnel, we don't know. We may be able to detect multiple genes, but mental illness may now be thought, and this is a slide prepared last year. So now the estimate is 8,000 genes are involved. Half your genome carries risk factors for mental illness. So what are you going to do with all that? I mean, there's nothing you can. So we could discover several rare variants that are local in origin, family specific, clan specific, and that would be somehow produce a generic disease. And understanding all the various pathways by which disease is produced would be very interesting, because it is very, very essential that we understand the nature of human brain and its mind. Now, this is a photograph you may have seen. This is the Solvay Conference of 1927. 17 people in the photograph who were awarded the Nobel Prize. 7 of those are known to have schizophrenia themselves or had a child or a parent with schizophrenia. So if you did genetic eugenic mapping, you'd be devoid of everything that we do right now. You won't be able to use a cell phone. You won't be able to build a satellite. You won't be able to do anything on Earth right now. You won't be able to do this talk unless you had people who had schizophrenia. So that is a fundamental problem. That is a fundamental problem. And as Spike Milligan very famously said, blessed are the cracked, for they let in the light. It's a very, very important thing to learn. So what we're talking about genetics is really the basis of what the great unknown world is. We can send satellites to the end of the universe, but we can't send a satellite to the depth of our consciousness. There is no way to do that. And to understand that process is really the crux of modern science, because this is completely unexplored, unknown territory. So thank you for your attention. These are all people who have collaborated. The most recent collaborators, like I said, there have been collaborators over the years from WashU, from IGIB, Ted Reich, who was a particular friend of Partho and mine, in CBS, in STEM, and CBS. And that's neuronal cell culture growing in the lab, thanks to Viju and Meera, who many of you know, and some of the younger colleagues, Dr. Reddy, and many others. So thank you all for your patient hearing. Thank you very much for this honor. I think this is a better closing slide than the last one, so I'll keep it at that.
So,
Over there.
Thanks very much, Professor Jain, for this extremely illuminating and intriguing talk. Now I would request Professor Parthapratim Majumdar again to invite Professor Sanjeev Jain to launch our NIBMG newsletter and also to felicitate the speaker.
Sanjeev. You have to type in NIBMG. Sounds more.
For the file?
Yeah.
It's done. Only one, two, one. No less.
What's inside? Where's the table of contents? You can just scroll down.
Nice. Right.
I haven't seen it. What's inside?
When the researchers do outside the lab, that sounds exciting. Right. Oh, it's clearer. There we are.
Now request Dr. Priyoshi Basu to kindly deliver the vote of thanks.
Good afternoon. Our guest of honor, Professor Sanjeev Jain, Director NIB Professor Taru Sharma, our founder director, Professor Partha Majumdar, ex-directors, Professor Shomila Sengupta and Professor Soumitra Das, our officiating director, Professor Arindam Moitro, our associate director, Professor Anupam Basu, my colleagues and staff of NIBMG, and my dear students. It is my honor to deliver the vote of thanks on today's auspicious occasion. It has been my privilege to have been present in all the Foundation Day celebrations in NIBMG so far, starting from 2011, when the guests of honor were Professor M.K. Bhan and Professor Obaid Siddiqui, and the keynote address was delivered by Professor Eric Green of National Institute of Health. So this was held in EZCC in Salt Lake, Kolkata, as we did not have a auditorium back then. And I remember that it rained so much that day that we were all standing outside in the courtyard, waiting for the guests to arrive to escort them into the hall. So we have come a long way since then. Today, I would like to express my heartiest gratitude to Professor Sanjeev Jain for such a wonderful and enlightening talk. We will cherish the memory for many, many years to come. Professor Jain has had a long-standing association with NIBMG. He was a member of the first scientific advisory committee of NIPMG. I would also like to thank the members of NIPMG who worked tirelessly to make this special day a success, especially our administration department, finance department, and our IT cell. Finally, I would like to thank you all for being here and making today a success. I hope you have enjoyed such a wonderful day. I know I did. And all the best for the future and have a wonderful weekend. Thank you.
So we have come to an end of the first half of the program, and lunch will be served in the canteen. So see you at 2:30 again for the debate competition. Yeah, thank you.
I group problem is there. It's not so yet.